So our last speaker for this morning is Nathalie Feiner. Morning. She studied and did her PhD in Konstanz, Germany with Axel Meyer, and then did postdocs with Peter Holland in Oxford, and then with Tobias Uller in Lund. And there she now leads her own group with ERC funding and other funding. She's going to talk about lizards, integrating, I think, a bunch of ecological, developmental, and maybe some phylogenetic aspects.
Okay, hello. Thank you for fixing the presentation. Thanks for the introduction, and thanks for the invitation to be here and present my research. So I will talk about Mediterranean wall lizards. So a main goal of evolutionary biology is to explain and understand adaptation. And adaptation is fundamentally a multivariate problem. So if you look, for example, at these seahorses here, if you can spot them, then you can appreciate that they need the right combination of behavior, morphology, and coloration in order to blend into the environment and make adaptation work. So in other words, adaptation requires phenotypic integration. And crypsis is a prime example of this, but it's not the only example. So adaptation generally requires phenotypic integration. And from an evolutionary perspective, we can ask a number of questions here. So for example, what is the evolutionary origin of trait integration? How is this trait integration maintained? And how can it be changed over evolutionary timescales? And if you think about what causes trait integration in the first place, what probably first comes to mind is correlational selection. So there's one classic example of correlational selection that I will explain you here. So these garter snakes in the United States, they come in different variation in their color, dorsal color patterns. So they can be very striped or they can be more blotched. And there's also variation in their escape behavior when they escape from predators. So they can just dash away or they can do many reversals when they slither away. And there's a tight correlation between these escape behaviors, and only snakes that have the right combination of dorsal color pattern with the right escape behavior have a high fitness and a high survival. So this is really correlational selection in action. And why this can be seen as sort of an external factor that causes trait integration, there's also internal factors that can cause it. So these are covariabilities. And covariabilities mean that traits are already integrated when they are produced during development. The most obvious example of this are supergenes. So the example here are these rough birds that come in different morphs. And the genes that regulate these different traits that go to these morphs, they are physically located in the same location on the genome. And often they sit in inversions, making them tightly correlated and being co-inherited. So therefore, you have this tight package of correlated traits that are inherited as one. But covariability can also be a little bit more subtle. So for example, when you have pleiotropies, and pleiotropies can occur in many different biological levels. Obvious level is the genetic level, when there's transcription factors, for example, that influence many other downstream target genes. At the physiological level, when hormones influence many other biological functions, and at the developmental level, when cell types give rise to many other cell types, and therefore you can have knock-on effects that link different traits together. And although I present these two here as alternatives, obviously they are intertwined, and the selective side is influencing the generative side and vice versa. So these are really fundamentally two sides of the same coin. So I've given you a little bit of a conceptual background here to phenotypic integration. And the rest of this talk I'm going to spend on talking about the evolutionary genetics and the ecology of phenotypic integration in wall lizards. In this talk, I'm going to focus on one species, that's the common wall lizards, Podarcis muralis, which has a very broad distribution across Europe. So it occurs from western areas of Spain until Turkey in the east. And throughout most of that distribution, these lizards look like this. So brown, slender, relatively generic lizards with not many exciting features. But if you go to central Italy and look at lizards of the very same species, then you see that there's a quite drastic difference in appearance. And if we compare the two types, then you see what's mostly conspicuous to your eyes is the coloration differences. So these green lizards here, they have UV blue spots on the side. They have a green dorsal coloration and the black ventral coloration. They differ also in morphological features, so they can reach up to twice the size in body size. And they have massive heads with long and wide skulls. And when they co-occur, they also show behavioral differences. So the green ones are socially dominant over the brown ones, and they show much higher levels of aggression. So we have systematic differences in coloration, morphology, but also behavior. And the obvious question that we are trying to answer here is, how did this ancestral type turn into this derived green type? And although these pictures might give you the impression that we are dealing with two different morphs, actually they occur on a continuous scale. And to make this point, I show you here a map of the sampling that we have conducted in the species. And I make the point by showing you the greenness, so the dorsal coloration here, but I could show you any other trait and you would get the same picture. So dorsal coloration, we score it in spectrophotometric data, but we can also score it simply by having a scale from 1 being entirely brown and 10 being entirely green animals. And then we have scored this for 70 plus populations across Italy. And I plotted here the average for males for each population. And then you can see that there's a nice continuous distribution across the landscape. If I would plot the same data for females, it would be slightly less green, but you would get the same pattern, basically. So the phenotypes are indeed continuous, and they vary across the landscape. And at least to some extent, this distribution here, with very, very green populations in around today's Rome, still green across the coast and stretching in along the coast of Liguria, but then slightly more attenuated in the inland areas. At least some of this variation is explained by climate, and that becomes obvious if I show you the altitude. So the Apennine Mountains seem to present a barrier for the greenness that doesn't get over this barrier. So this greenness here is strongest here and is spreading over the coast of Liguria up almost until the border to France. And if you know something about the phylogeography of the species, then this might come as a surprise. If you don't, then I'll show you here. So the species falls out into 6 deep phylogenetic lineages. And the study area that we're dealing here with, the northern Italian area, is actually the region where the oldest lineage, the blue lineage, the Southern Alps lineage, is meeting the central Italian lineage. And the divergence time between these two lineages is 6 million years old. So it's a really, really old divergence, and the species, or these lineages rather, within one species are in secondary contact exactly in our study region. So now you can appreciate that it's actually quite surprising that the phenotype, the greenness here, is not only present within one lineage, but it has spread over a hybrid zone into a second lineage as well. So with this context in mind, now we can ask because if we would be dealing with a syndrome, what I supposed in the beginning, then we would find a pairwise correlation between the individual traits of the syndrome. And that is indeed the case. So when we here look at 5 different traits, 2 morphological and 3 coloration traits, we really find within the Italian lineage, on the population level, we find a very tight correlation pairwise between these traits. And even in the hybrid zone, where these traits are introgressing into, even there we find a statistically significant correlation. Although it's slightly weaker, the level of correlation, but it's still statistically significant and not significantly weaker than in the Italian main lineage. So what this tells us that the syndrome, so the tight integration between individual traits in the syndrome, it's maintained even in the face of introgression over this hybrid zone. And we're dealing indeed with a real syndrome with tight integration between the traits. So, but where does this syndrome come from? What's the origin? And with origin, I mean now geographically speaking. So to answer this question, we used the following design. So we selected individuals from 6 geographic regions, so from pure green, pure brown regions, and we did this within the Italian lineage for 2 different transects, Rome and Tuscany, and within the Southern Alps lineage. So this is the lineage that's 6 million years divergent from this lineage. And again, here we selected pure green and pure browns. And then we generated RAD-seq data. Anne has just explained what RAD-seq data is. So it's a reduced representation of the genome. And then we built phylogenetic trees. If we build a phylogenetic tree of the entire genome-wide data set, then we get basically a clustering by geographic regions. So it's simply isolation by distance. But if we restrict the analysis to the loci that are associated with the syndrome traits, so it's roughly 5% that are associated with the syndrome traits, then we get a clustering according to phenotypes. So then the green individuals fall out as monophyletic. And based on the topology of this tree, we can infer that the geographic origin of this green syndrome was around today's Rome. And from there, the phenotypes have spread along the coast. As I said, they don't make it over the Apennine Mountains, but they spread along the coast, even into the Southern Alps distantly related lineage. So why is the syndrome spreading? What does it make successful and outcompete other lizards? To understand what's driving the spread of the syndrome, we conducted a series of experiments. Most of these experiments included these outdoor enclosures. So these are 10 by 10 meter outdoor enclosures with these bruise blocks that lizards love to bask on. And we had 10 of these outdoor enclosures. And what we did for a couple of years is that we populated these outdoor enclosures with either pure brown and pure green lizards or with a continuous variation of lizards. And we populated them during spring, let them do what lizards do for a couple of months. And after they have been mated, we brought the lizards back into the lab, let the females lay their eggs, and then based on the eggs, we could score hatching success and paternity for each of the eggs. And what we also got from these experiments is, of course, behavioral observations, because we had students stationed around these enclosures that could observe these individually marked lizards and see who's hanging out with whom, and use this to reconstruct social networks between them as well. And we coupled this with mate choice experiments in the lab to really get the full picture of the intraspecific interactions within these lizards. I cut a long story short because the results are very clear. What we found, number one, we found that within males, the syndrome traits are associated with higher reproductive success. So the higher a male scores on the blackness or on the lateral blue spots scale, the more offspring they would reproduce. Number two, we found that in experimental contact zones, green males are dominant over brown males. And even, so this is body size, and this is brown males, and this is green males, and even relatively small brown males would be dominant over, even small green males would be dominant over brown males. And this higher dominance would translate into higher success in courtship, so actually higher success with mating with females. And number three, we found that in experimental contact zones, so when we had brown and pure greens within one enclosure, then we found that the mating is assortative, so meaning that the green females would predominantly have offspring with green males and brown females predominantly with brown fathers. But there's also asymmetry, meaning that green females almost never mated with brown fathers, but brown females would almost have 40% of their offspring sired by green males. So there's a lot of asymmetry in this system. So what we conclude from this is that sexual selection is mediated by male-male competition, and there's an absence of female mate choice, and that this sexual selection is actually driving the asymmetric gene flow that's pushing the green phenotypes into this distantly related lineage in the hybrid zone. And from this, we can generate prediction about gene flow in the hybrid zone, which holds up remarkably well, and is published in this paper. So, finally, what is the genomic basis of the syndrome? So to answer this question, it was very helpful to have the introgression in the hybrid zone, because what that actually constitutes is like natural outcross experiment, and it gives us a very high power to identify actually causal loci of the syndrome. The design that we adopted here was that we used the same setting as before. So we had pure green and pure brown for 3 different paired transects. And for 60 individuals across this design, we generated whole genome resequence data, and then we did FST outlier scans in paired analysis here. I show you the results here visualized first as a Manhattan plot. So we analyzed the data in each lineage specifically. So what we found is that there's a number of peaks of association that you see. Many of the peaks are specific for each lineage, but we also find a number of peaks that are shared between the 2 lineages. And that's what we would expect, because we would expect a shared origin and a shared genomic basis between the 2 lineages. If we look closer into these 86 genomic regions that are shared outliers between the lineages, and these 86 regions are associated with 86 genes, then we find that a bit more than half of them are actually annotated. So these are the ones that we can draw biological meaningful information from. None of these 47 annotated genes is an obvious coloration gene. And we've also collected a big transcriptomic data set where we looked at transcriptomic differences between different colored skin patches. And none of the genes is popping up in these analyses. So we don't think that we have found any coloration gene per se. But a bit more of half of these genes, so 28 genes, have a link to neurocrest cell biology. That got me very excited. Might not be obvious why, because you might think that these different traits have probably nothing in common. Coloration is based on pigment cells in the skin. The differences in heads is based on cartilage and bones in the skeleton of the skull. And the behavior is probably based on hormonal differences or neurological differences. But what all these traits actually share is that they develop from the same developmental cell type, and that's neurocrest cells. So neurocrest cells are a vertebrate-specific cell type. They originate in the early embryo when the neural tube closes at the crest of the neural tube, therefore the name. And these cells are interesting because they migrate out from the neural tube. They migrate out into different areas of the embryo, and then they differentiate and give rise to many different cell types. And these cell types are pigment cells in the skin. They are the bone and cartilage forming cells in the skull, and they are these adrenal gland cells and nerval cells of the peripheral nervous system. So these cell types have clearly something to do with the coloration, morphology, and behavior that our lizards differ so strongly in. And what you can easily appreciate from this scheme here is that if you change anything in the regulation of neurocrest cells early in development, for example, increased proliferation or increased migration, then this will have knock-on effects on all these cell types together. So you have this inherent covariability between these traits due to their shared origin in neurocrest cells. And this shared covariability was recognized by Adam Wilkins, and he suggested that this might be the explanation for the domestication syndrome that has been observed already by Darwin in our domesticated animals. And so the fact that half of our outlier genes are indeed neurocrest cell-like regulators is very suggestive, and we at the moment we are following this up with more developmental cell biology to test if this really is the case. So, but we didn't stop our genomic analysis here, because FST outlier scans are, of course, a quite blunt tool. So we wanted to use more data and more approaches to refine our results. So what we have done is that we've, in addition to these FST outlier scans, we've used these 60 whole genome resequenced genomes to also look at Tajima's D, because we had predictions for differences in genetic diversity. And we also complemented this with an independent data set on 900 individuals for which we had generated RAD-seq data, and we conducted a genome-wide association study on these 900 individuals. And here, as a response variable, we used the first principal component of all syndrome traits. And since all of them are correlated really well, this PC1 has a very high explanatory power for the phenotypes that we're dealing with. So when we run all these analysis independently, and when we then intersect between them and ask which are the genomic regions that pop up as outliers consistently across all analyses in all lineages, then we find a single genomic region. This is one on chromosome 12. And if we zoom into this region in a bit more detail, then we can see that the region consists actually of two different peaks with very high association with the syndrome. And these peaks are lying on they're very narrow peaks, so they lie on two different genes, RAB18 and ACBD5. Both of them do make sense from a neurocrest cell hypothesis perspective, because especially RAB18, is a known regulator of cell migration, so it does make a lot of sense. But what's interesting here is that we have this peculiar architecture with a region, it's a 300 kb region in between, that does not seem to be associated with the syndrome, but it's nevertheless very interesting because it has these duplicated genes sitting here. So we have 6 copies of a gene called patched and 5 copies of a gene called PKS. And these duplicates that we find here are not what we find in the lizards in our Italian study system, but this is the reference genome. And the reference genome comes from an individual from Spain, so it is from a relatively distant related lineage. So we actually don't exactly know what's going on in our Italian lineage in this region, but the Spanish reference genome has this interesting duplication patterns here. And we looked a little bit closer in the gene trees of these genes, and what we found is that they are a mixture of very old and very new duplicates. So some duplicates date back to the origin of reptiles, and some duplicates are probably species-specific to our wall lizards. So we've done a lot of analysis to get better insight into what's going on here in our Italian lizards, and I show you just one visually striking result that gives you an idea. So here we plotted the cumulative read depth across all our 60 reference genome, and the red dashed line indicates what you would expect if the genomic region would be present just once, or actually twice, diploid within the data set. And you can see that this is all fine outside and within these peaks, but within, in this interpeak region, we find a mixture of copy number variation with several duplicates, but also a loss probably with some regions that are probably not covered by reads at least, so they're probably not present. So this interpeak region really shows signs of copy number variation in our Italian lizards. To understand better what's going on in this region, we decided to develop some new genomic resources. And here I will cut a very long story very short. So what we've done is that we selected an individual from a pure green population and an individual from a pure brown population that are genetically very similar, but phenotypically drastically different. And we reconstructed 2 new reference genomes for them by using a combination of long-read sequencing and high-C for assembly. And we also used optical maps. And what we ended up with is 2 new reference genomes that are of excellent quality. They are to chromosome scale. Everything is entirely perfect, except for our candidate region. So we're really glad that we have done a sequence-independent approach, which are optical maps, because this tells us that we have assembled everything perfectly, but there's a gap in our candidate region. And what these optical maps have taught us is that if you're not familiar with optical maps data, then I would just walk you through now. So this is the optical maps of these 2 new reference genomes. So it's for the exact same individuals as we prepared the reference genome, we also prepared optical maps. And then we align these optical maps with the Spanish reference genome here in our candidate regions. And these gray lines means that there's an alignment, so these regions can be aligned between these optical maps to the reference genome. But within this repetitive region, we can't align these optical maps. And if you look at these repeat sequences, then you can also say that it's a pattern of a lot of short motifs that are duplicated, but also inverted many times differently. And the fact that we find 2 optical maps means that our 2 individuals are heterozygotes, so they have 2 different alleles of these structural variations. And the fact that they have different, so we have 4 different alleles here from 2 different individuals, that shows that the structural variation is segregating in the Italian population. And we're still trying to figure out exactly what this region has to do with these peaks that we see flanking this repetitive region. Okay, I just briefly sum up the key results that I presented to you. So I've shown you that the phenotypes that we're dealing with here, they are continuously varying across the landscape, but the traits go together, so it's really a syndrome. I showed you that sexual selection is driving the introgressive spread of the syndrome into this distantly related lineage. And I've shown you that the genomic basis is rather complex, but that we have a large proportion of these outlier genes that seem to regulate neurocrest cell behavior. And I've also shown you that there are structural rearrangements that likely maintain an evolutionary labile hotspot of variation. At least that's our interpretation so far. A brief outlook is that we're continuing right now with more developmental biology to really understand neurocrest cell biology and to see if there's a difference in neurocrest cell biology between the pure greens and the pure browns. We're doing that by sorting neurocrest cells from early embryos and then combining that with single-cell RNA sequencing. And we're also expanding the comparison to other wall-lizard species because today I focused on 1 species only, but at least in 5 other wall-lizard species across the Mediterranean, it seems that this syndrome has evolved. So we're right now doing a lot of field work and trying to document phenotypes in great detail to quantify if the syndrome is really repeatedly exactly the same or in how much they differ between different species. This was, of course, a team effort. So the whole project was started by Tobias and Jeff more than 10 years ago. And Young was the postdoc who have done a lot of the early genomic analysis. And I'm also grateful to all the collaborators and team members that have contributed over the years to this project. Thanks. I'm happy to take questions.